Hello everyone, today I'm going to show you how I cultured the cells for the semester. So what I did was I put collagen 1.5 mils inside of each well, and I left it in the incubator at 37 degrees Celsius overnight. And now I'm aspirating that. So you're getting rid of the liquid. You put the pasture pipette, this little glass pipette is a pasture pipette. You put it straight down to the bottom of each well, and then that way you get rid of the liquid, but you don't disturb the matrix, the collagen 1 matrix at the bottom of the dish. And then you immediately close your dish. This is part of the aseptic technique. You only have things open as you need them immediately. You don't just keep them open. So now I'm setting up my station. That tissue on the right hand side is a Kim wipe, and I spray it with ethanol, and then I leave it on the right hand side in case I have any spills. So here in each of those 15 mil conical tubes, I added 4 or 5 mils of media, and then this is the first cryo vial. And I think those are MCECs. Those are mouse cardiac endothelial cells. And normally we would transfer these into the media and centrifuge them and then resuspend the pellet in clean media. But here, the company said not to do that. So I'm just taking a little bit of clean media. I'm adding it into the cryo vial drop-wise. Don't let the tip of the touch anything, like don't let the tip touch anything, because otherwise you can risk contamination. So then I added it dropwise, and then I'm resuspending, and then I'm transferring it to that 15 mil conical tube. So my goal is to get these frozen cells, and I want to homogenize them within that 5 mils of fresh media, because this way the DMSO that is inside of the cells right now, it gets diluted, because normally DMSO is toxic to cells at room temperature. So we want to dilute that before we seed the cells. So here I'm just resuspending to make sure that the cells become homogeneous within the mixture. Okay, so notice another thing, that I'm never, once something is open, I never put my hand over it, because I can risk, like if there's any bacteria or fungus or little threads on my coat, I can accidentally drop those into the cell suspension, and that can cause contamination. So anytime something is open, I try to crawl with my hand around the hood. And so here I'm using a serological pipette. These big plastic ones are serological pipettes. And so this is what I'm going to use to resuspend the cells to homogenize them, but also to seed my dish. They apply less pressure. When seeding, remember that we don't want to apply too much pressure to the bottom of the dish when we seed, because we don't want to disturb that collagen 1 at the bottom. It took us a whole 24 hours to get that set up nicely so that the cells have something to attach to. And so now I'm just adding 2.5 mils of the cell suspension into each of 2 wells. Okay, now I've gotten the other cryo vial, and I think these are MAECs or something. Mouse aortic endothelial cells. So it's a different cell type. One of these cell types is immortalized, and so that means that it has been transformed so that it can divide almost an unlimited number of times. so that one vial we could just expand over and over and over again for years. And then the other cell type is a primary cell type, and those will only divide about 5 times according to the company. So once you've divided 5 times, then you would want to purchase more cells. So yeah, I'm just performing the exact same steps, and notice like the aseptic technique, how good it is. I'm being so careful not to touch anything with the tip of the pipette. And then I'm working, I think I'm working slowly because I don't want to risk contamination. I don't want to risk going over that 15 mil conical tube. Again, here's the serological pipette. You should definitely start learning the names of things, and that's why I'm going to be very repetitive in all my videos. And that's the 15 mil conical tube. I'm resuspending my cells, and then I'm just going to seed half of them in one well and then half of them in the other well. So normally we would use a larger plate to seed, but because I'm thawing fresh for the first time, I don't know how many of these cells are alive, so usually you want to increase your seeding density. So if you would normally seed like 10,000 cells per centimeter squared, I would go and do like 15 or 20,000 just to make sure that you have like enough cells that actually attach to the bottom and survive. Okay, so I went ahead and added some 70% ethanol to my Kim wipe, and then I went ahead and cleaned my area. And now, what am I doing? I think I'm labeling the dish or something. Oh, I wanted to show you. Okay, normally when you seed, you want to slowly go left and right, and then backward and forward with your dish. And you want to do that for about 2 minutes when you first, oh, trash. Okay. So here I'm just showing you where you're supposed to toss all of the different items, and you have to do it correctly.